Hello, I'm Isabella Grumbach, University of Oklahoma, Vice Chair of this meeting. I'm here with Dr. Joseph Wu, the Director of the Stanford Cardiovascular Institute. This morning, we had an absolutely stunning plenary session on frontiers and hot topics in cardiovascular medicine. And Dr. Wu started this session with a really breathtaking whirlwind tour of things to come: genomics, NAMs, AI in drug discovery. You started out with some examples of how you're using the screening of available libraries with your iPSC workflows, and you've been able to define and discover some already approved compounds and medications, for example, to treat cardiac fibrosis and now also pulmonary fibrosis. Where do you see the next steps when it comes to vascular disease?
Yeah, so thank you so much, Isabella, for inviting me here. As I mentioned on the talk, we focus mostly on 4 pillars of human-centric biology, and that is human iPS cells, CRISPR editing, genomics, and an AI-based drug discovery. I think the 4 pillars allows you to discover or rediscover or repurpose drugs that could be used for fibrosis indications. I gave 2 examples. One is for artesunate, and I think the other one is for seralutinib. You could also use it for inflammation example. One example I gave is genestin, which is actually a isoflavonone found in soybean, and it has a pleiotropic effect in terms of dampening inflammation. It's supposedly one of the reasons why some people claim Japanese people live longer, because they have a much higher soy diet in their food intake compared to Western diet. There are multiple examples that you could go do using this NAMs approach. And I think in the future, it's really up to your imagination to see if you want to take iPSL-based macrophages, iPSL differentiated T cells or B cells, and then study them for your project. I think the key point I try to emphasize is that now you have access to all these patients with different diseases. It's much better to start your front end of your biological discovery research by looking at the heterogeneity of these patients and try to narrow down on which one is your highest bet in terms of your therapeutic target. And this is otherwise not possible if you use a traditional single cell line or traditional one string animal, in which you're already locked into one string or already locked into one species.
You also introduced the concept of NAMs, and not everybody in the audience was familiar with these novel approach methodologies. And I understand you've worked with the FDA, and you also gave us an overview of how the FDA and other regulatory agencies are really interested in these novel approaches to replace animal work. And you mentioned this, one of the really amazing opportunities here is that if you test 25 different strains of iPSCs, you do take heterogeneity into account, whereas in animal studies where we have usually inbred mice, we don't. Are there any other opportunities, advantages of NAMs that you would like to highlight for us?
Yeah, so NAMs refers to new approach methodologies. It really came about back around 2022, when the FDA Modernization Act 2.0 says, hey, can we replace or can we decrease the reliance on animal models and switch it to more of a human-centric biology, human-centric stem cells or organoids? Many people, including our lab, we've been working on it for the past 20 years, right? Ever since the discovery of human iPS cell. Back then, when we look at human iPS cell, discovered back in 2006, you quickly realized that, hey, I could generate iPS cells from everybody in my hospital and differentiate to their patient-specific brain organoid, differentiate to their patient-specific liver organoid, differentiate to patient-specific cardio organoids. And so the beauty is really starting with a human-centric biology, human-centric target. Now, what I would say is there's a conflicting interest between academia and also drug discovery, meaning that in academia, we want to have a tight p-value. the postdocs, the graduate students want to graduate, get a paper published. So it's much easier to take a single cell line and pick a cell line that gives you nice p-value difference with a very tight standard deviation. Right? That's been happening over the past 100 years. That's how we do science. But if you're going to bet $100 million on a drug, you don't want that. You basically want to de-risk the whole drug discovery portfolio by taking your top 2, 3, 4, 5 compounds and at a fraction of the cost, put it on 100, put it on 500 iPS cell lines at a time. And just kind of give you a rough sense of which one of your drug can consistently hit the targets that you want. And I think, as I mentioned in my talk, I gave the example of nicotine. If you give nicotine to iPS cell-derived endothelial cells, in our study among 20 patients, 12 male, 8 female, there were probably about 10 of them that responded. The other 10 did not respond that well. Similarly, I gave an example of a tyrosine kinase inhibitor, sunitinib, in which you give it sunitinib to iPS cell-derived cardiomyocytes in 20 patients. Some people will respond and some people will not respond. So you're trying to look for a drug in which it can consistently see it. It's not going to be 100% because no drug worked that well. But if you can see your positive signal in 50, 60%, that's pretty good for you to go ahead with investing $100 million in a phase 2, phase 3 trial. And so that's what I've been trying to advocate and trying to, to the FDA and also to the NIH and also to our academic colleagues, that how we do drug discovery in the past, relying on a single strain of mouse or single cell line, it makes no sense. Yeah.
You also discussed the related and concept of clinical trials in a dish, and you gave us an example of a family with dilated cardiomyopathy and a lamin A mutation. So this is really a beautiful way of discovering treatments for rare diseases. Where do you see opportunities in vascular disease? Vascular disease tends to be a little messier. There are different cell types. The diseases take quite long to develop, and often there's shear stress and other factors. Where do you see are the first steps for clinical trials in addition?
I think the clinical trial in addition, CTID, I've been saying this CTID acronym for the past 10, 15 years. I think the earliest entry or the easiest entry of this clinical trial in this concept will be looking at patients with rare orphan disease that are related to vascular diseases. But one example will be progeria. I think there's an FDA-approved drug called lorlatinib, and in our lab, we're trying to say, besides lorlatinib, are there other drugs that we could screen using progeria's patients' iPS cell-derived endothelial cells? Other examples you could go after is HST, patients with hereditary hemorrhagic telangiectasia, fibromuscular disease, patients with idiopathic pulmonary fibrosis, in which it's more of a fibrosis-related disease, but there's also some immune vascular component. There are many examples of how you could do this in a low-hanging fruit by going after these well-characterized genetic diseases. Once you have established a proof of principle and once you have confidence of how do you set this up, then you could go to a larger scale, for example, looking at more of a polygenic diseases in vascular complications. Yeah.
We also had a question from the audience about the differentiation of iPSCs. And I understand for example, when it comes to endothelial cells, there is quite some variability among the cells that you differentiate in a dish. What are the frontiers? What are the experimental challenges to overcome before we can take the next steps?
So during my talk, I kind of highlighted maybe about 10 areas of research for the young trainees to focus on. One example is tissue maturation versus throughput versus scalability versus integration with the host organ system versus non-invasive imaging and universal media. So multiple areas. And one of the hurdles of iPSL is that when we differentiate these iPSL cardiomyocytes or iPSL endothelial cells or iPSL smooth muscle cells, they are more of a neonatal stage. There are many labs, again, including us, that are trying to develop protocols to make it more into a mature state. It's never going to be the same as an adult mature cell, because adult mature cell is growing in the presence of a 3D milieu in the body. Because if you take an adult mature cell, if you take an adult liver cell, or adult endothelial cell, or adult fibroblast, day 1 or day 2, it looks the same, but then in a dish condition, by day 7, it would have de-differentiated. So you never make it the same, but you'll make it closer, and that's what a lot of people are trying to do. I do think it's just a matter of biology, understanding the differentiation biology, and I'll give an example. Back in 2004, we were working with 2004, 22 years ago, we were working with human embryonic stem cell, and we were trying to differentiate human embryonic stem cells into beating cardiomyocytes. At that time, we were not using monolayer. We were using what we call a hanging drop, in which it's inverted, and then the embryo body twitch. And when we see it twitch, we're dumping our chest, because if it twitch, it means we got cardiomyocyte. And within that embryo body, we're getting about 1, 2, 3% cardiomyocytes, and that's it. Fast forward 22 years later, the CVI, the Stanford Cardiovascular Institute, we have summer programs in which we have high school students who come by for 8 weeks. And within 8 weeks, the high school students have already become a master of differentiating cardiomyocytes. They're getting 95% pure cardiomyocytes. And so that just tells you how fast the technology improves. So the question of making it more mature, making it more aged-like, I think that's going to be solved. And it's also part of the reason why during the talk, I mentioned that we've routinely give out these cell lines. We've given out more than 4,000 vials to more than 450 investigators, because I want more and more investigators to work on this model, and collectively, we in the scientific community, with our minds out together, can push the field even faster.
The field has come an incredibly long way in the last few years. Looking forward, where will we be in 5 years?
Yeah, so in 5 years, I would hope that the concept of NAMS, NAMS, and the concept of CTID, clinical trial and dish, these 2 acronyms become a regular vocabulary for our trainees and our postdocs. Right now, it's still not a common vocabulary yet, but hopefully in 5 years it will. And it's got to have a fundamental shift, right? Because both the NIH and the FDA is advocating for it. And that means the new generation of trainees that are coming in will get trained in this area. And then 10 years, 15 years from now, you're going to have many, many more people who are working on it. And so we work with a lot of senior PIs. We also work with some pharmaceutical companies, in which we actually have to hold their hands and train them and teach them how to do it. But I do think that 5 to 10 years from now, this will not be a problem. Everybody will know how do you generate organoids. Everybody will know how do you differentiate different cell types, and how do you do the standardized drug screening assay. And because everybody's working on it, then science will move much faster.
Thank you very much for your introduction to this really exciting and fast-moving area, and I'm sure we will have you back in a few years so that we will hear all the updates. Thank you.
Thank you, Isabella. Thank you for inviting me here. And I hope everybody have a great time at the Vascular Discovery meeting here in Seattle. Thank you.